information on this project in a short amount of time, enough to keep you interested, but not enough so I can tease you and have you come over to my poster and ask some more questions. So I'm Will. I'm an MD PhD student at OHSU, which is located in Portland, Oregon, which is in the northwest of the United States, if you haven't visited before. And I am excited to tell you more about my method in predicting transcription factor activity using prior biological information. Now, this may be a concept that's familiar to most of you, but just so we're all on the same page, transcription factors coordinate cellular gene expression programs. They do this by binding to promoters or enhancer regions throughout the genome and enabling the transcription of RNA and ultimately the protein translation. And the composition of the different proteins that are translated help dictate the cellular phenotype. And a lot of evidence has been shown that this process is fundamentally dysregulated disease, that either having mutations at in the gene themselves or on the transcription factor binding site leads to dysregulation of these proteins and ultimately a disease phenotype, in this case a leukemic cancer cell. And there's also been evidence showing that overactivation of this process or overamplification of certain RNA transcripts can lead to a disease phenotype. This study, specifically Emek, has been shown, we're currently implicated in different cancer types, that its overamplification is associated with cancer development. And so the central question that we ask in our project is, how can we develop a tool to detect aberrant transcription factor activity which may be driving disease? And so we turn to a method or a database that our group has helped curate called Pathway Commons, which curates peer-reviewed information on biological pathways, including gene regulatory networks. I am happy to go into more information about how this works, but it uses natural language processing to glean information from publications, and currently has information on over 5,700 pathways and 2.2 million interactions over 22 high-quality databases. And so what Priori does is, from an RNA sequencing data set, it calculates the relative activity of a transcription factor. What it first does is it pulls the target genes for a given transcription factor in a database from Pathway Commons, and takes the Spearman correlation between the expression of the transcription factor and its downstream target genes to calculate a weight associated with that pair. And it takes two pieces of information from that weight. The sign of the Spearman correlation gives an indication of the direction of transcriptional regulation, whereas the degree of the Spearman correlation coefficient gives an idea of the impact of transcriptional regulation of that transcription factor on its target gene. So we use the sign of the weight to divide up the target genes into two groups, rank their relative expressions, and then the score is just the summation of the product of the weights and their relative ranks. Very straightforward, especially compared to some of the other methods that have been presented. But we wanted to take this methodology and see how well it performed to other methods, because the idea of looking at transcription factor activity is not necessarily new, but there's been a lot of controversy in it, and then being able to produce models that reflect known biological systems. So we made use of this decoupler pipeline, which does exactly this. It takes advantage of a data set curated by Holland et al., which includes 124 knockdown and overexpression data sets. So these are cell lines where a certain gene has been knocked down with CRISPR-Cas9, short hairpin RNA, or overexpressed a gene using a TET-inducible system, followed by RNA sequencing. And then using decoupler, we can generate activity scores for these different methods, including Priori, and look at the frequency in which the perturbed transcription factor score is among the top scores compared to the unperturbed. Ideally, you would see the perturbed transcription factor being among the top relative to the other ones. And we can give a summary of the sensitivity and specificity that this method identifies the perturbed transcription factor using the area under the precision recall curve or the receiver operating curve. And when we do this, we were really encouraged to see that Priori's AUPRC and AUROC was much greater than the other methods that we tested, indicating that it had a much greater degree of sensitivity and specificity than these other methods. And this was also consistent with the results that decoupler found in their publication of this workflow. So we were encouraged in the reproducibility of this workflow. And so we applied this method to 2 different databases to see if we could find known or define drivers of disease in breast cancer, as well as mediators of drug sensitivity in leukemia. And we generated activity scores for 637 patients from the TCGA bracketed data set. Each one of these points is a dimensional reduction of a patient's entire activity scores. And you can see that they very cleanly cluster into their basal and luminal molecular subtypes. And when we also repeated this exercise for the top 3 other activity methods that were nominated from the decoupler analysis, and there's no clear delineation between the molecular subtypes when we perform the same dimensional reduction. We performed clustering once again and looked at the patterns in these transcription factor activity scores to make sure that we see what we would expect. Taking a step back, these molecular subtypes, basal and luminal cancers, are phenotypically differentiated by the expression of their hormone receptors, specifically progesterone receptor and estrogen receptor, which are encoded by NR3C1 and ESR1. And in the population associated with triple negative cancer, you see a decrease in those activities consistent with what we would expect, as well as a decrease in GATA3 activity, which is associated with luminal cell specification, and an increase in MYC expression, MYC activity, which has been shown in basal cancers. But the transcription factor that stood out to us was FOXA1, which is a fork head protein, as it was it's the greatest different activity between these 2 clusters. And so if you normally differentiate these patient populations by subtype, you don't see a difference in survival. However, if you differentiate them by their FOXA1 activity scores, you see there's a significant difference, giving some indication that these findings are clinically relevant. But importantly, if you instead differentiate them by FOXA1 expression, the survival difference goes away, indicating that the activity, including the expression of the transcription of FOXA1's target genes, is really important for understanding this clinical disease. And we can repeat this exercise again with these top 3 other methods, differentiating by their own activity scores for FOXA1, and we don't see any survival differences. 1 minute. All right, I'll try to go through this quickly. So we also wanted to see if we can do this, but for mediators of drug sensitivity. So we leveraged the Beat-AML database, which has RNA sequencing data and matched ex vivo drug sensitivity data. So each one of these dots is the correlation between drug sensitivity and transcription factor activity. And one that stood out to us was that FOXO1, another fork head protein, was strongly associated with venetoclax resistance. And which is interesting because when we did repeated with the other methods, we saw that only norm W mean agreed with this hypothesis, whereas Viper and ORA said that it was associated with venetoclax sensitivity, giving us an imminently testable hypothesis. So when we knocked down FOXO1 in monocytic AML leukemia cell lines, we saw that it sensitized these cells to leukemia consistent with priori and norm W means hypothesis. I want to take a second to thank my mentorship team as well as the co-authors of my paper and my other lab members that I work with.
I assume it's known how FOXA1 is activated. I'm just a bit confused that you get like this nice kind of patient stratification using your predicted activity scores, but then it vanishes when you use FOXA1 expression, right? Are there alternative modes that you could get from whatever TCGA data, proteomics data or something?
Are you talking? Sorry. Yeah, so are you talking, because the FOXA1 expression is only looking at its normalized counts, but that activity score has baked in the downstream target genes of those expression. What you could do is pull out the weights that you use. You can pull out the weights of the activity score and find the strongest drivers of that activity and use those to nominate as these might be the strongest drivers of this activity, and those can be further investigated as a potential network that might be dictating survival. And I do that a little bit in the preprint, and it's on the poster, but not enough time to show it. Yeah.
Any other questions? Okay. I guess I have a question. So the activity is based basically on expression values, is that right? So of course, it could be also DNA methylation or accessibility, and so the fact that the transcription factor is in the cell doesn't necessarily mean that it's binding to your binding site.
Yep.
So could you actually use this method to identify, so where you have low area under the curve, to identify potential transcription factors that are methylation specific, for example? Or?
Well, so to directly answer your question, so I do humid work. So this method was actually developed because we were trying to find transcription factor regulators that dictated the mechanism of a drug combination. And what we did is we correlated the activity scores in this BetaML database with the expression of a gene that we were interested in. And we used that to find transcription factor STAT5 in this case that we thought was an important regulator. And when we went in and actually altered the expression of STAT5, we did see important changes in that gene expression. So to indirectly answer your question, yes, you can use this method to nominate and hypothesize important transcription factor regulators. I don't know much about hypomethylation. I guess you would have to be very thoughtful about if you're looking at methylation signal or important regulators in methylation. Yeah, what we targeted. Yeah, of course.
Okay, so if there are no further questions, we're moving on to the next speaker.
Thank you.
Elimde tutsam olur mu? Bunu elimde tutsam olur mu? Hi, my name is Ozgun Babur. It's my pleasure to introduce our recent public nucleic acid research publication, inferring transcription programs driven by somatic alterations using interpretable deep learning. This is a joint work from a previous PhD student from the Russell Schwartz group and Sojun from my group. As you know, mutations, copy number alterations, and epigenetic modifications deregulate signaling pathways and change the gene expression programs. And transcription factors are main link between this upstream signaling and downstream transcriptional programs. In cancer, one of the main questions in cancer is whether the mutations and copy number variations impact on transcription programs are similar or different between different cancer types. This may inform new therapeutic avenues for cancer patients. Here we utilize somatic alteration, ataxic data, epigenetic chromatin accessibility data, as well as gene expression data to model this flow of information. When we integrate these three different data sources, we are inferring patient-specific transcription factor activities, then associating them with the mutation as well as copy number alteration profile of the patients. Then we can ask different questions, whether the mutations impact or similar or different between different cancer types. this is how formal we are doing this. We develop an interpretable deep learning approach called Citrus, chromatin-informed inference of transcriptional regulators using self-attention mechanism. Input to our computational framework is the somatic alteration data. And we are encoding this to tumor embedding layer using the self-attention mechanism. We also use cancer type input to account the heterogeneity between different cancer types. Then in the middle layer, we are transforming this tumor embedding layer, the transcription factor layer, where we are going to retrieve the patient-specific transcription factor activities. We also use ataxic data to get more context-specific transcription factor target genes using a motif search. And we use ataxic data to connect this transcription factor layer to target gene expression. Basically, in the last layer, we are predicting the gene expression from this inferred TF activity layer and transcription factor target priors based on chromatin accessibility data. When we model this flow of information, we are learning attention weights for each alteration, which is giving the importance of it predicting the gene expression, as well as patient-specific transcription factor activities. Let me show you how we are going to use these two outputs from this model to better understand the impact of somatic alterations. In this figure, I'm showing you the predicting attention weights from the model in y-axis and frequency of those somatic alterations in the x-axis. You can see that there is a correlation between attention weights as well as frequencies. For example, like TP53, PIK3CA, highly frequent genes across many different cancer types are getting high attention weights. But there are other somatic alterations, although they are frequent, their attention weight scores are lower. When we perform an enrichment analysis, we saw that genes have high attention weight are enriched with the cancer drivers. In this figure, I'm showing you the inferred TF activity from the model. Every dot represents one patient, and patients are colored by the cancer type. Mostly, when we look at this cluster, we are seeing the patients are clustering within their cancer type, but we are also seeing that cancers related to each other, for example, adenoma carcinomas are clustering together, or cancers from the same tissue are clustering together. For example, kidney cancers are clustering together. Main goal of this project was like understanding impact of somatic alterations on transcription programs. Like what are the disrupted transcription factor downstream of a specific mutation or copy number variation. As a proof of principle, I'm just giving you one example today from the breast cancer. In breast cancer, like PIK3CA, PI, and GATA3 is frequently altered, and in this figure, I am summarizing the transcription factors associated with these alterations using our model. Let's take one example, PIK3CA. PIK3CA has activating mutations, and it deregulates the AKT PIK3 pathway, which is, has many different functions such as cell proliferation, also crosstalk with other pathways. When we look at the transcription factors associated with PIK3CA in breast cancer, we are seeing, for example, HIF1 alpha, and this is already known association. It is downstream of the AKT pathway, but we are also identifying new associations such as STAT4 and MFAT-C1. You can see that when there is a activating mutation, STAT4 and MFAT-C1 activity becomes higher compared to wild-type cases, and these two transcription factors are involved in immune pathway. When you do this analysis using the mRNA levels of these transcription factors, you don't see the distinction between wild-type and mutant patients. Actually, like late last year, there was a publication where they generate epigenomic and transcriptomic data between mutant and wild-type breast cancer mouse as well as cell line models, and they show that like difference between the transcription factors STAT4 and MFAT-C between wild-type and mutant cases. In in summary, CITRIS is a deep learning model to model the impact of somatic alterations on transcription programs in a pan-cancer manner to gain mechanistic insights. And by using the cell attention mechanism, we were able to capture nonlinear effects of mutations on gene expression programs. And using patient ataxic data allow us to get more context-specific gene and transcription factor relationships. And this framework allows us to also quantify effect of transcription factors which are normally obscured by their modest mRNA levels. Today I just give you a brief overview of the paper. If you want to check more details, you can look at our recent publication as well as our GitHub page. This work, as I mentioned, led by Yifeng Tao, a former PhD student from the Russell Schwartz group, who is now a quantitative researcher at Stadel Securities, as well as a software engineer, Sojuma, from my group. And I want to also thank NCI for funding this research. And thank you for listening. I'm happy to take any questions.
Thank you very much. Lovely talk. Any questions?
Yes, please. Question. What method did you use for interpretation of your model? What method did you use for interpretation of your model when you did?
So when we are interpreting our model, we use literature resources, whether our predictions are recapitulating what is already done. Mm-hmm.
Okay. Other questions?
Yeah. Can you show us the architecture? So the deep part, the deep part is essentially the embedding of the
Yes.
cancer types. The rest is shallow. That's why you can interpret it, right?
So
The TF layer is just one layer, and then you associate it with TFs.
yes, actually I'm not giving many details. Actually here we are also using ensemble approach. As you know, these activities that we are learning, the ways that we are learning is not stable. So we are using the ensemble of models to summarize the activity per patient. Mm-hmm.
Okay. Thank you very much. More questions?
Sure, I'll ask it. I couldn't help noticing on your attention weights that the single nucleotide variants look very good. The copy number aberrations look like a little bit of a who's who of suspicious copy number genes. I wonder if you think they're real or if there's some artifact or not.
It's tricky to encode copy number alterations into the model because they are more also amplification like heterozygous application or homozygous application. So it is tricky. And here we are just making the binary.
So do you believe the ones you're seeing, or do you think that?
Case by cases, like when we look at this attention weight figure per cancer type, like for example, colorectal cancer, like there's a copy number alterations APC, and we were able to see the high attention weight.
Thank you again.
Thank you.